Hi, I'm Sarah Maurer, and today we're going to talk about how protocells can be used as a model for the origins of life. Protocells are really important for first organisms because they are aggregates that can serve to co-localize and protect metabolic and genetic components, and they can also be used to define an individual, which is necessary within a population for Darwinian selection to occur. And then the other thing that aggregates can really do is interact with the metabolism. So not only are membranes necessary for things like electron transport and for defining chemical gradients within a cell, but aggregates can also be used to catalyze reactions directly. And we see this when we think about how fast a reaction will occur in a hydrophobic phase, like in a protein pocket, versus in a hydrophilic phase, like straight water. When we talk about aggregates that are used to model the origins of life, they really fall into 3, maybe 4 categories. The first category is the one that looks most like a cell, which we often call vesicles, or is sometimes referred to as a liposome. And a vesicle is a clear membrane bilayer that has an aqueous interior and an aqueous exterior, but a hydrophobic barrier that serves to separate the interior contents from the exterior contents. We can also have something like an oil droplet, which has a hydrophobic core, and then it usually has amphiphiles on the outside that help to stabilize the oil droplet, something like salad dressing. And then the last type of aggregate that's often talked about when we talk about the origins of life is called a coacervate. And while oil droplets may be sometimes considered coacervates, generally when we mention a coacervate, what we mean is that you take a cationic or a positively charged polymer and you mix it with some negatively charged molecules, and they tend to clump up into something that looks like an oil droplet, but instead of being hydrophobic on the inside or water hating, instead it is highly charged. And so anything that's highly charged can sequester on the insides of these coacervate structures. Maybe the last type of structure that we talk about when we talk about origins of life are inorganic structures. So things like very thin films of inorganic material that can serve as a barrier between maybe 2 water systems like you might see in a hydrothermal vent. Or even maybe something more like a crystal, which can serve to co-localize material on the surface, but could be an individual in a population if the crystals were very small and there were many of them, like grains of sand. And so these self-assembled structures can serve to aggregate materials, including genetic and metabolic components, and to identify an individual for selection. So what affects aggregation of these different molecules? Of course, the concentration of those materials that are aggregating is going to play a huge role in aggregation. So if you don't have enough cationic polymers, you're not going to form coacervates. If you don't have enough amphiphiles, you're not going to form bilayers. So you're going to need to have a sufficient concentration of your materials that are forming these structures first. Once you have that concentration, then you can start to think about other factors that affect aggregation, like temperature. High temperatures tend to cause things to dissolve more easily, to become more soluble in water. Low temperatures tend to cause things to precipitate or freeze. We can also have things like ionic strength of the solution affecting whether or not things aggregate. Too low of an ionic strength and things might be highly soluble. Too high of an ionic strength and things precipitate out. And then we can also change the pH of our systems. We've talked about hydrothermal vents, which often have vastly different pHs from ocean water. And so the origins of life may have needed to suffer through pretty extreme pH changes in its transition to becoming modern life. And so when we think about aggregation, usually we pick aggregates that are stable under the conditions that we want them to form. But for any environment, so high pH, high temperature, we can usually find some kind of aggregate that can form that will serve as a good model for our origins of life chemistry. What chemistry do protocells generally harbor? Well, you can think of aggregates as interacting with the molecules that are going to start life in a few different ways. In the cartoon you see here, you can see that in the A scenario, molecules are interacting with the surface of our aggregate through something like electrostatic interactions or perhaps even hydrogen bonding interactions. We can also have the hydrophobic effects sequester non-polar materials into our hydrophobic bilayer, or if we have an oil droplet, into the center of a hydrophobic oil droplet like you see in B. In C, you can see an amphiphilic compound that is anchored into our hydrophobic core, but also has a portion of it exposed to the environment. And then finally, we can have, at least with liposomes and sometimes with coacervates, a molecule that is sequestered on the inside of our aggregate, but is still hydrated. It's still in a water phase. So here you see these little triangles that are in the interior of our membrane. So what do these membranes really do to our prebiotic reactions? We can have two types of reaction. The first is a membrane-associated reaction, or a surface-associated reaction. So in these reactions, S is the substrate, shown in green. Our product is black, and presumably the product would be useful to the protocell in some way. Maybe it stabilizes the structure, maybe it's a metabolic or a genetic component that can then catalyze new reactions. And additionally, we expect that some of the products of the reaction are not useful, and so we call those waste. The benefit of a surface-driven reaction is that the waste can just diffuse away into the environment, while the products and the substrates are associated pretty strongly with our bilayer, maybe through electrostatic interactions or hydrophobic interactions. If we have a full vesicle with our catalytic network encapsulated in the interior, then our substrate first needs to diffuse through the hydrophobic barrier, where it can be turned on the interior, where it can be turned into products. And then additionally, if there are waste products made, then these waste products are going to have to diffuse away out through the hydrophobic barrier again. And so these are kind of the two mechanisms that we use to model prebiotic reactions. An example of one such reaction is RNA enzyme called the hammerhead ribozyme. The hammerhead ribozyme can self-cleave. If you see, there's a little red arrow that points to the CU connection at the node in the center. And the hammerhead ribozyme cuts between the cytosine and the uracil here. And we can actually monitor the rate of reaction inside of our membranes. And what we find is that with no vesicles present, we have an apparent rate of about 0.5. When we encapsulate the ribozyme within vesicles, we actually see that the rate is not quite as fast. The self-cleaving doesn't happen as quickly. Our rate decreases by about half. And this is actually kind of expected, because when you constrain your environment, if the waste products can't diffuse away, then you're going to limit the rate of reaction, because the waste products are still in your reactant, possibly inhibiting it. Interestingly enough, if you do the same reaction in a coacervate, the reaction increases pretty significantly in your coacervate aggregate. And part of the reason for that is that the coacervate concentrates your cleaved strand substrate with your catalytic enzymatic uncleaved strand. And so being able to concentrate your reactants really can speed up your reaction. Protocells are also capable of growth and division, which means replication in a very simple sense. And if you look at the cartoon shown here, it's been shown that if you have a reaction that is growing and increasing the osmolarity of your solution, that membrane will actually grow larger by stealing molecules from a non-growing vesicle. In the movie that you're seeing here, we're actually watching the transition of an oil droplet into vesicle phases. So you can see that there are little buds that kind of start to appear on the surface of our oil droplets. And then those buds slowly, as the motion of the fluid in the system, will wash the membranes away from our oil droplets. And so we really do have this natural growth and division process, this self-assembly that is totally independent of any genetically controlled mechanism, but that would be controlled by environmental forces like wind and water flow through a system. So when we think about the evolution of our protocells, essentially we start with some kind of prebiotic soup. And aggregation happens so that we form these self-assembled protocells that have some lifelike properties, but maybe are not fully alive. Through some natural processes, maybe some people like to call it chemical evolution, we end up decreasing our molecular diversity of our prebiotic soup, But at the same time, we increase our functional complexity by selection processes and by iterative rounds of replication of our protocells. This leads to first life, which is a much simpler form of life than we expect to see in modern organisms. And that first life will then undergo true Darwinian evolution with information polymers and maybe even a second type of polymer like protein, generating and undergoing information phylogenetic relationship to generate LUCA, what we think of as LUCA. And this LUCA would then turn into all modern life. And so this really is how protocells can fit into our origin of life model and help us to better understand the evolution of our early Earth environments.